Embryo development starts with one cell and ends up with billions of cells that make up living beings like us. There are many different types of cells. For example, skin cells, blood cells, muscle cells, brain cells, etc. And these cells organize into tissues. It is a great challenge for biology to understand the process we call cell differentiation that leads to the specialization and to the organization of cells during embryo development. There will be two parts in my talk. In the first part, I will describe the experimental evidence supporting a model of cell differentiation based on stochastic, meaning random, gene expression. And in the second part, I will present the result of the computer simulation of the same model. Classically, cells are believed to follow the instructions of a deterministic genetic program. I have proposed a different model according to which cells first engage in cell differentiation through stochastic gene expression, then stabilize through selective interactions. This model is called the Darwinian Model of cell differentiation because of its analogy to natural selection. However, the term selection here is taken in a loose sense. It can mean the death of cells, or more generally, their selective stabilization. This diagram here on the slide illustrates the general principle of the model with the simplest case. In a cell, just to illustrate the general principle, in a cell, two genes, A or B, can be stochastically activated, either A or B. And according to which gene is activated, the cell either becomes a type A cell or a type B cell. And in a cell population, it is the interaction between the two types of cell that leads to the stabilization of stochastic gene expression and to the stabilization of their type. A theoretical model must allow for experimental prediction. A theoretical model is judged on its capacity to make experimental predictions that must be verified. The Darwinian model allowed for a first major prediction to be made in the early 1980s when it was originally proposed. Gene expression is stochastic. At that time, the idea that gene expression is deterministic following the mono-Jacob was overwhelmingly prevailing. Today, stochastic gene expression is an established fact. But this prediction could be made even more accurate. Indeed, if the model is correct, one expects to observe a peak in the variability of gene expression, a surge in the variability of gene expression preceding cell differentiation and followed by a decay. This is illustrated in this picture that is extracted from the thesis of a student who did his PhD with me 20 years ago. The colored discs at the top of the slide illustrates symbolized cells expressing genes stochastically, thus producing variable and unstable gene expression patterns and unstable potential phenotypes. And among these cells, only the black and red one can stabilize through their selective interaction. So, bottom of the slide, if one quantifies and the variability of gene expression, one expects to observe a peak of variability corresponding to the phase of stochastic gene expression, followed by a decay corresponding to the phase of selection. So the experiment was done by Olivier Gendrillon and his team, with whom I collaborated. 92 differentially expressed genes, meaning genes that either become expressed or repressed during cell differentiation, were analyzed in 96 chicken blood differentiating cells. As can be seen on panel A, 8 hours after cells were triggered to differentiate, we observed a peak in the variability. So this curve shows the average variability of gene expression at different time points. And as expected, as predicted, 8 hours after cells were triggered, we observed a peak of variability followed by a decay. This curve is perfectly reproducible, and the differences between time points are reproducible, are statistically significant. Moreover, this result was confirmed on the right of the slide by the analysis of the number of significant correlations in expression between two genes at the different time points after cells were triggered. And again, as expected, 8 hours after cells were triggered, we observed a decrease in the number of significant correlation, followed by an increase, thus confirming the initial surge in the variability, in the disorder in gene expression. The same phenomenon, the same peak in gene expression variability was observed in another cell lines in human blood cells by Andras Spaldi and his team. In their study, they not only analyzed gene expression, but they also followed cells under a microscope and they filmed cells. The division history and the morphological change of these cells that had been triggered to differentiate was recorded. Globally, they observed a great heterogeneity of behavior of each cell. Notably, cells switch randomly phenotype before becoming stably differentiated, stably committed to a differentiation pathway. So I'm going to show you a movie of this experiment, and you're going to see cells that are triggered to differentiate, and that instead of following a straightforward pathway toward differentiation, hesitate between different types. One cell has switched to round. You can easily distinguish undifferentiated cells are polarized, whereas differentiating cells are round. So they switch back and forth between the two morphologies. At one stage, they will divide. They will be, now one cell has divided. They are all round. And now they are round, and they switch back. One has switched back to the undifferentiated type. After several cell cycles, a fraction of these cells will become stably committed to the differentiation pathway. So this experiment shows visually, you have seen it with your own eyes, that cells do not follow a uniform path. They do not follow a program toward differentiation. On the contrary, randomness is inherent to the process. Moreover, the same result, the same phenomenon, the peak of variability, was observed in another lab in America, in Sui Wang's lab, and it was also detected in a series of other cell lines, notably in in different mouse embryonic cells. Thus, this phenomenon is not exceptional. This is not an exception. On the contrary, it appears to be rather a general feature of cell differentiation. So now I'm going to switch to the second part of my talk and present the result of the computer simulation of this model of cell differentiation. I did this work in collaboration with Bertrand Laforge, who is here, who is a physicist at the University of Paris, and who will talk later this morning. So computer simulation can be done in various ways with different goals. Our initial goal when we did this work was to evaluate the relevance of the Darwinian model of cell differentiation as a theoretical framework to understand cell differentiation. Therefore, the question we wanted to answer was, can the Darwinian model generate tissue organization? And more generally, what would be the properties of a cell population obeying its rules? So in this study, we want to test the intrinsic properties of the model. We are not modeling a real system. So we used cellular automata, and we started with the simplest possible model. In the simulations I'm about to show you, cells adapt to their internal environment by switching type randomly. And when they are adapted, they proliferate, and through their proliferation, through their activity, they produce molecules that stabilize them. So in this simulation, I need to give a little bit of details. In this simulation, so you can follow on the slide, in this simulation, a cell is a square that multiplies in a two-dimensional matrix, and there are two types of cells, red and green. Red cells correspond to the activity of a red gene and synthesize red molecules. Similarly, green cells correspond to the activity of a green gene and synthesize green molecules. And these molecules diffuse in the environment of cells, that is, in the matrix in which the simulation takes place. Red and green genes are expressed stochastically, thus cells can switch randomly type. Red cells can become green, green can become red, with a switching probability that depends on the concentration of the red and green molecules they produce, in the concentration of these molecules in the environment. These molecules are equivalent to pleiotropic factors, which are common in biology, that can have multiple effects. First, cells are independent for proliferation. This is a selective part of the model. These molecules act as growth factors, meaning red cells need green molecules to survive and proliferate, and similarly, green cells need red molecules to survive and proliferate. Second, stabilization. This is a stabilization part of the model. Cells are stabilized by their own activity, that is, by their own product. And this stabilization occurs according to a sigmoid function, starting with a switching probability, green to red or red to green, equal to 1, when the concentration of the molecules is null. This probability decreases until, as the concentration of molecules increases due to the activity of cells, until this probability is null and the cells are stabilized. Red cells are stabilized by their own product, red molecules. Similarly, green cells are stabilized by green molecules. There are 12 parameters eventually in the model that control molecules turnover, diffusion, cell division, cell death, probability of cell type switching. We first ran the model by varying the quantitative values of the parameters of the model. And as you can see on the slide, we observed several shapes, several cellular patterns, masses of interspaced red and green cells, alternating larger areas of red and green cells, even rings of cells. And these patterns, these forms, these shapes were constant, were reproducible as long as the parameters, values of the model were kept constant. So these are other examples. And we became particularly interested, and in fact we focused all our attention in one of these structures, this one on the left shown by the arrow, because it shows clear organization. It is a regular cell bilayer made of 2 layers of cell, 1 layer of red cells, 1 layer of green cells of constant width, and these layers are all adjacent, and the growth of this structure is finite. It never invades the whole matrix, even if the simulation is kept running indefinitely. The panels, the picture on the left of the slide, show a typical bilayer formation during a simulation run. Panel A, starting with 16 cells chosen randomly. The cell bilayer, panel B, first gets nucleated. As the simulation continues, panel C, the cell bilayer grows longitudinally. It looks like a sort of worm. So the magic space here is a torus, meaning that opposite sides are in fact adjacent. So the structure continues growing, panel D, until it buckles. Once it has buckled, it stops growing. Somehow it has reached, the worm has reached its adult stage. It won't change anymore, even if the simulation is kept running indefinitely. Each time the simulation is run again, the same cell bilayer organization is always reproduced. Top right of the slide are 2 other examples. The shape of the worm, of the structure, may vary longitudinally, but the typical cell bilayer structure, pattern, organization is always reproduced. Thus, the simulation shows that cells obeying the rules of the Darwinian model have the potential to generate a reproducible tissue organization of finite growth. We next wanted to understand why is growth finite in this structure. This was quite an unexpected result for us, because when we started this work, we were thinking of tissue organization, but not really of the control of cell proliferation. In the computer program, there is no explicit command, stop command, saying to the cell, stop dividing. So we analyzed the distribution of the molecules, red and green, across a lateral section of the cell bilayer, as shown by the arrow on the top right of the slide. And we realized that on the 2 edges of the cell bilayer, on the edges of the cell bilayer, 2 conditions are met that cause growth arrest. So this is shown at the bottom of the slide. This is the edge of the red layer, and this is the edge of the green layer. So on these edges, 2 conditions are met. On the one hand, here on the red edge, the concentration of growth factor, meaning green molecules for red cells, is so low that these cells can't proliferate. But this is not enough. At the same time, on the other hand, because these cells could switch to green phenotype and resume growth, as happened in the other case of computer simulation. At the same time, the concentration of red molecules here is so high that the switching probability of these cells is null. They are totally stabilized, and they can't switch to the other type. So it is the conjunction of these 2 factors that create an equilibrium state of the structure, and the structure is fixed. This equilibrium state obviously depends on the quantitative values of the model parameters. This can be easily shown by changing one by one the values of these parameters. In this example shown on the slide, we altered the value of a parameter, beta, which is involved, which controls the switching probability of cells. As can be seen, so cell stabilization was altered, was destroyed. The cell bilayer was destroyed, as can be seen, with cells proliferating indefinitely and invading the whole matrix. Same result occurred, we obtained the same result when another parameter, L, that controls the diffusion of molecules, was altered. The interdependence of cells, the interactive selection of cells was altered, and as a result, the cell bilayer was destroyed with cells proliferating indefinitely and invading the whole matrix. Thus, organizational properties and the control of cell proliferation come from a combination of specific values of the model parameters which create an equilibrium state. But ultimately, all the parameters are either involved in stochastic gene expression or in the selective stabilization of cells. Therefore, tissue organization is an equilibrium state between stochastic gene expression and the selective stabilization of these cells in the context of this model. So in this experiment that I have just shown you, we had drastically altered the values of the parameters. We next proceeded to much smaller changes, to slight deviations in the values of these parameters. And we found limit values corresponding to states close to equilibrium in which the proliferation of cells resumes locally at specific positions without the overall structure of the cell bilayer of the worm being destroyed. So this is shown on this slide. In panel A, we first let the cell bilayer, the worm, form and reach its adult stage. Then we introduced a slight deviation in one of the parameters. And as can be seen on panel B, cell proliferation resumed locally, as shown by the two arrows, with cells budding out of the cell bilayer. As the simulation continued, these cells, panel C at the bottom left, these cells grew into cell masses, panel D, that were eventually released in the environment. Thus, it seems that the cell bilayer, our little worm, was in a state resembling cancer with localized, uncontrolled cell proliferation. These slight deviations in the values of the parameters in real cells, they could correspond either to mutations that would modify the properties of a protein, or more simply to the interaction of a protein with a carcinogen product that would also modify the biophysical properties of this protein, thus either altering the interaction with DNA and gene expression, or altering its diffusion, thus altering the repartition of molecules in the whole living being and the interaction and the cellular environment. That would result in a disturbance of this equilibrium between stochastic gene expression and selective stabilization, which in turn would lead to localized cell proliferation. So I'm going to show you a film of this computer simulation experiment. In the first part of the film, you will see the cell bilayer formation. We let it grow until it reached its adult stage. At that stage, when we did the experiment, we introduced a slight deviation in one of the parameters, and as you will see, cells resumed proliferation locally at specific location, but the overall structure of the worm remains intact. So you see now the cell bilayer forms, and it continues growing until it reaches its adult stage. Not yet. It continues growing. Now it has reached its adult stage. So we introduced a slight deviation. It would not change anymore if we do nothing. We introduced a slight change, and you see cell proliferation resumes locally, budding out of the cell structure, but the cell bilayer, the worm, remains intact. In conclusion, the experiments that I have shown you, both the experiment performed in real cells and the computer simulation experiments confirm the predictions of the Darwinian model. They show that, cells do not use disorder, not information, in order to differentiate.
On sait que la notion de biologie telle qu'on l'entend aujourd'hui, et le mot même a été fondé au début du 19e siècle, et que le 19e siècle, c'est le siècle où s'installent petit à petit les disciplines, tout du moins leur naissance telle qu'on les connaît après, dans le 20e siècle et aujourd'hui. Et assez rapidement, en fait, j'ai l'impression que ces disciplines s'organisent sur un axe évolutionnaire, notamment à partir du paradigme darwinien au milieu du siècle, et sur un axe plus physiologique, pensons au programme de la théorie cellulaire qui se met en place sur tout le 19e siècle, et avec notre figure nationale de Claude Bernard, qui a eu le grand mérite notamment de mettre en place la notion de milieu intérieur. Si je veux mettre en avant ces deux paradigmes, évolutionnaire et physiologique de l'autre côté, c'est pour dire que Darwin, avant l'aspect physiologique, avait permis de montrer que la téléologie à l'œuvre dans la nature était aveugle. Or, j'ai l'impression que cette séparation, cette structuration du champ des différentes disciplines biologiques entre, d'un côté, donc, les domaines évolutionnaires et de l'autre côté, les domaines physiologiques, ont mis du côté physiologique un certain retard dans cette manière d'appréhender la théologie à l'œuvre dans le vivant. Or, et c'est là la force de votre exposé, c'est de montrer que finalement cette espèce de téléologie aveugle que le modèle darwinien avait mis en avant bien avant, il est aussi à l'œuvre dans les phénomènes de la physiologie. Et j'ai l'impression que c'est quand même un point majeur de votre exposé, de montrer qu'il y a vraiment une puissance créative de la stochasticité à l'œuvre dans les phénomènes physiologiques cellulaires, notamment donc, selon les paramètres initiaux, et qui crée finalement une variabilité initiale extrêmement riche. Finalement, cette variabilité initiale donne une sorte de vertige, en témoigne la question qu'on a eue par rapport aux généticiens au milieu de la génétique. Et pourtant, ce vertige, vous l'avez montré, est totalement appréhendable par des modèles, notamment des modèles à partir de différents automates cellulaires. Donc finalement, en fait, tout en étant fondé sur des phénomènes stochastiques, on arrive quand même à des formes d'équilibre transitoire. Et finalement, ces équilibres transitoires peuvent nous faire croire, dans une forme de mouvement un peu a posteriori, encore à une téléologie à l'œuvre. On n'arrive pas finalement à s'en sortir. Mais, et c'est là aussi encore une force de votre exposé, c'est de montrer qu'il ne faut pas oublier que quand bien même ces modèles nous permettent d'appréhender de nouveau ces phénomènes stochastiques, ils peuvent aussi surprendre eux-mêmes. Vous l'avez dit, notamment dans le développement de ces modèles, vous cherchiez à cerner la question de la différenciation cellulaire. Or, vous vous êtes rendu compte de l'importance du poids de la prolifération et de la croissance des cellules. Donc finalement, en fait, outre votre exposé qui permet vraiment de montrer à quel point la téléologie est forte en biologie et qu'il faut vraiment réussir à s'en départir, il montre aussi que l'efficacité des modèles n'est pas à prendre à la légère parce que, outre la capacité de rendre compte du monde, elle permet aussi de l'appréhender sous un nouveau point de vue et un point de vue créateur. Il y a vraiment une force des modèles. Donc vraiment, merci beaucoup pour cet exposé. On peut le remercier de nouveau, Jean-Jacques Cupiec. Merci.